Today I am taking up part 3 of sexual reproduction in flowering plants, and it deals with pollen pistil interactions and double fertilization. First, let us take up pollen pistil interactions. In the previous class I have told you, as the gametes are immobile in the plants, they need some pollinating agents which are either animals or wind, water, etc. Animate or inanimate agents. Pollen grains occur in various forms, various shapes, various colors, and various texture. And the pollen grains will be falling upon the stigma. Pollen grains of various types of plants will be coming into contact with the stigma, and the stigma of a plant has to identify the right type of pollen grain. For this purpose, the stigma interacts with the pollen grain through a chemical dialogue. Dialogue means it is the discussion between two persons, but here it is the chemical dialogue between the pollen grain and the stigma of a plant, stigma of a pistil. When the pollen grain lands upon the stigma, due to this chemical dialogue, the stigma identifies whether the pollen grain is of a compatible one or non-compatible one. If it is of right type, then the stigma accepts the pollen grain. Otherwise, the pollen grain is rejected and it will not germinate. Or even if it germinates, pollen tube is not formed. So, if a right type of pollen grain falls upon the stigma, stigma secretes sucrose solution which promotes the germination of the pollen grain and formation of the pollen tube through one of the germ ports. I have told you in the previous class that the pollen grains are released either in two-celled state or three-celled state. If the pollen grains are released in two-celled state, the pollen grain contains one male gamete or one germinative nucleus and one vegetative cell. If the pollen grains are shed in three-celled state, it has two male gametes and one vegetative cell. If the pollen grains are in two-celled state, what happens is, in the stigma itself, the male gamete undergoes mitotic division and produces two male gametes. In this way, three cells are formed, two male gametes and one vegetative cell. Then, as the pollen tube grows through style, the male gametes and the vegetative cell also travel through the pollen tube. The pollen tube, after passing through style, enters the ovum and passes through the micropyle and enters one of the synergid cells. I have told you in the previous class, this is the micropolar end having two synergid cells and one ovum. The opposite end is known as the antipodal end and it has three antipodal cells. In the middle of the ovum, there are two polar nuclei, free polar nuclei, and the cell is known as central cell. The synergid cells have filiform apparatus. The function of the filiform apparatus is to guide the pollen tube into the synergid cell, into one of the synergid cells. After entering the synergid cells, the pollen tube ruptures and releases the two male gametes. These are two male gametes. The vegetative cell degenerates. Now, one of the important event occurs in fertilization. So there are two male gametes and only one ovum. One of the male gamete fuses with the ovum. This fusion is known as syngamy. Both the ovum and male gamete have only haploid number of chromosomes, that is n number of chromosomes. Through syngamy, a zygote is formed, and in the zygote, the diploid number of chromosomes is restored. So in this way, zygote is formed. In future, the zygote develops into the embryo. So the embryo has diploid number of chromosomes. The other male gamete fuses with the two free nuclei present in the central cell. Here, three nuclei are fusing. Three haploid nuclei are undergoing fusion. This is known as triple fusion. Because of the triple fusion, what happens? 3n number of chromosomes are formed. The nucleus formed through triple fusion is known as primary endosperm nucleus or PEN. The cell, the central cell, becomes the primary endosperm cell. The primary endosperm cell produces in future the endosperm. Now, we have to learn one important term here. Double fertilization. In all the flowering plants, double fertilization takes place. Double fertilization means fertilization of the ovum and fertilization of the free nuclei with the male gametes. That is, there is two types of fertilization. One fertilization produces the zygote. Other type of fertilization produces primary endosperm cell. So in this way, two types of fertilization takes place in the flowering plants. That's why it is known as double fertilization. And double fertilization is the important feature of all flowering plants. So in this way, syngamy and triple fusion takes place. Then, the embryonic development begins. The zygote develops and also the endosperm develops. But here, the development of endosperm precedes the development of the zygote. Why? Because the endosperm has to provide nutrition to the growing embryo. So, the development of the endosperm begins first. Generally, what happens is the earlier development of endosperm is of free nuclear division type. That means thousands of free nuclei are formed first. Only then the cell walls are formed. In this way, free nuclear divisions takes place first in the endosperm. One good example for this is tender coconut water. The tender coconut water is nothing but free endosperm nuclei. The fluid contains thousands of daughter nuclei. Only the white kernel present around the fluid is the cellular endosperm. Then, after the formation of the endosperm, the developing embryo consumes the endosperm and grows. In some plants like pea, groundnut, beans, etc., the endosperm is completely consumed by the developing embryo. So, no more endosperm is seen in the seeds. But in plants like coconut, castor, etc., the endosperm is consumed only partially. So, the remnants of endosperm are still found in the seeds. So, after initial, or here we can remember one more point, the embryonic development of both monocotyledons and dicotyledons is similar in the beginning, or the initial embryonic development in both monocotyledons and dicotyledons is identical. The zygote develops first into pre-embryo, proembryo. The proembryo develops into a globular embryo, then it develops into a heart-shaped embryo, and finally it develops into a mature embryo. In this state, the seeds are formed and they are shed. The integuments of the ovule forms the seed coats, and the seed coats protect the embryo from desiccation and from other problems. Then, when the conditions are right, water and air passes through the micropyle and the seeds germinate into a plant. And one more point, after fertilization, double fertilization, what happens? The synergids degenerate. Similarly, the antipodal cells also degenerate. Only the primary endosperm cell and the zygote develops into future embryos. Then, in the mature embryo, we find one embryonal axis. This is the embryonal axis. Here it is the embryonal axis. In the case of dicotyledons, two cotyledons are present. In the case of monocotyledons, as the name implies, a single cotyledon is present. The single cotyledon present in the monocots is known as a scutellum. In the dicots, these are the cotyledons. The embryonal axis present above the level of cotyledons is known as epicotyle. The epicotyle develops into the future shoot system. The embryonal axis present below the cotyledons is known as the radicle. The radicle gives rise to future root system. The radicle is covered by a root cap which is protective in nature. Coming to the monocotyledons, this is the scutellum. Here also the embryonal axis above the scutellum is known as epicotyle. The epicotyle is represented by rudimentary leaves, and the epicotyle is covered by a membrane known as coleoptile. Coleoptile is a protective structure above the epicotyle. This is the hypocotyle. Hypocotyle has a root cap, and the hypocotyle is also covered by coleorhiza. In this way, the seeds are shed in the case of monocotyledons and dicotyledons. In the next class, I will be dealing with some of the important terms associated with reproduction in flowering plants, and these points may be very important in the case of NEET examination. Thank you.